Hello, I'd like to thank Jay Wu and the Symposium for Inflammatory Skin Disease for the invitation to speak here today. My name is Brian Kim, and I'm Associate Professor of Dermatology at Washington University School of Medicine. I'll be talking today about itch and atopic dermatitis, and in doing so, I'll leverage a lot of the recent innovations in the biology of itch and how it relates to some of the findings in clinical trials and in the future, how we need to perhaps think about itch and the complexity of it within atopic dermatitis. Here are my disclosures. So we've been studying eczema, at least in my laboratory program, for over 10 years. And when we first embarked on these studies, there were a number of things that we knew about eczema, and there's a lot that's changed. So what was it like over 10 years ago? Well, it was certainly appreciated that it was a very high incidence disease, although the epidemiology on it was less clear. We knew that it disproportionately affected children, and topical steroids were the mainstay first-line therapeutic for this condition. There was limited knowledge about the immune mechanisms, and what I'll hopefully highlight is how this has changed. A central focus of our scientific program is understanding how healthy skin becomes inflamed skin. So in essence, what are the molecular and cellular mechanisms by which what we see as a rash in the skin is initiated and propagated? So when we first started studying atopic dermatitis, the original paradigm of this disease was really that there are inflammatory or allergenic stimuli in the environment that somehow through a damaged or impaired skin barrier results in the activation of an adaptive Th2 cell or T helper type 2 cell, characterized by the production of the Th2 cell associated cytokines IL-4 and IL-13. We know that this process results in the pathologic production of IgE, which I'll come back to later in the talk, as well as damage directly to the skin barrier itself. We, in recent years, discovered that rare circulating basophils actually come out of the blood and into the skin and are major sources of IL-4, particularly in lesional atopic dermatitis skin. And we also discovered for the first time previously unrecognized group 2 innate lymphoid cells, or ILC2s, shown here in orange, as major sources of IL-13 in the skin, and essentially expanding the paradigm in our understanding of atopic dermatitis beyond adaptive antigen-specific Th2 cells towards these innate cell populations, both basophils and ILC2s, as being major pathogenic drivers of atopic dermatitis through the production of these cytokines IL-4 and 13. We now know that, we now refer to these cytokines not necessarily as Th2 cell cytokines, but also just more broadly type 2 cytokines, appreciating the fact that there are a variety of innate sources beyond Th2 cells. We know that these cytokines are very pathologic, and in the real world, this has become very, very evident in the treatment of moderate to severe atopic dermatitis with the advent of dupilumab. When these studies were actually coming to the forefront, as dupilumab was only approved in 2017, it was well recognized by a variety of investigators that the itch in atopic dermatitis improves rather dramatically and in unprecedented manner. And that's when we started to become more interested in the itch in atopic dermatitis. Going back to the original slide, I talked about where we were at 10 years ago with eczema. Well, one thing that was notably missing was itch. And there was not a whole lot of focus previously when we first were studying eczema with regard to itch, and that has changed tremendously in recent years, and I'll highlight what's happened here. So now we're interested in understanding what are the molecular and cellular mechanisms that within the inflamed skin or the rash trigger pathologic sensory processes such as itch. To understand this, we have to actually understand better the what we call the somatosensory nervous system, which is the conscious perception of the stimuli from our environment, whether it may be various things like pressure that's mediated by these corpuscles here, frictional signals that may come from these A beta or A delta nerve fibers, and then shown here in red are non-peptidergic C fibers, which mediate a variety of sensations. The red fibers alone, whether it's pain, itch, temperature, and a variety of other sensations. When we hone in on these non-peptidergic red C fibers shown here, how does neurotransmission work? Well, we know classically that neurotransmitters bind their receptors. These will typically be G protein coupled receptors. And then these receptors actually then trigger the opening of cation channels. You get a cation channel gradient. And if you get a sufficient gradient that reaches a certain threshold, what happens is that voltage gadolinium sodium channels open, and then you get an electrical impulse known as an action potential being sent to the spinal cord. From the spinal cord, you get signals going up to the brain, and that's how we essentially perceive itch. And these are the basics of neurotransmission. Well, how does this work with a real world example? We can all relate to the molecule histamine. We know that if we inject histamine into our skin, it will bind its receptor, as an example here, H1R, which will be very much dependent on these cation channels referred to, such as TRPV1, which is the receptor for capsaicin, and a minority percentage of these neurons alone can mediate itch in response to histamine. One thing we know about histamine is that it's been somewhat disappointing in the clinic because the vast majority of chronic itch disorders do not respond to antihistamines, particularly in the context of atopic dermatitis. So, however, fortunately, new biology has given us a lot more hope that there may be other targets. One such pathway is the IL-31 pathway. Discovered in 2004, we know that if you overexpress IL-31 in mice, they'll itch. They can bind their receptors directly on sensory neurons, so the IL-31 can act actually as a neurotransmitter, and it's variably dependent also on these cation channels, and you get even a very small percentage of sensory neurons responding to IL-31 and sufficient to actually mediate itch. What we do know is that IL-31 derived from immune cells can trigger sensory neurons to mediate itch, and the signal works its way up to the brain. The question then is, what happens if you block IL-31? We know that this is a potential therapeutic strategy now. So in recent years, we actually asked a very simple question, which is, if IL-31 can do this, and there is an agent called nemolizumab that's in development and that can do this in human atopic dermatitis, we asked a very simple question in recent years, which is that with these innate cell populations being major sources of type 2 cytokines, can these classical type 2 cytokines function in a similar manner? And if we block this, can we have some effect on itch at the neuronal level? Well, this work I'm highlighting here was done by my graduate student, MD PhD student Landon Etchen, who discovered and shown here that in fact IL-4 receptor alpha, the target for dupilumab, is actually expressed within mouse dorsal root sensory ganglia, as well as a native IL-13 receptor. He could also readily pick up expression of IL-31 receptor, and this is very much conserved between both mice and humans, indicating that these type 2 cytokines, while their receptor is actually expressed within the sensory ganglia. So the question then is, do sensory neurons, if they express these receptors, actually respond to type 2 cytokines? We can test this by using a technique called calcium imaging, in which we can actually label neurons with dye that then change color or fluorescence upon influx of calcium, which indicates activation of the neurons. And then that's the fluorescence here is on the y-axis, on the x-axis is time. And what we find is that if we spike in recombinant IL-4 just in a matter of a couple minutes, we see a nice signal in response to IL-4. We can confirm that these are viable neurons by putting in positive control of potassium chloride. Human cadaveric donor DRG similarly respond to human recombinant IL-4. You see that there's a spike in fluorescence after a couple to few minutes, and then we can confirm that these neurons are actually responsive to capsaicin, indicating that in fact these are TRPV1 positive sensory neurons. The question then becomes, if these neurons actually express the receptors and that they're responsive, does this really matter at the end of the day? And the only way to understand this kind of itch biology is to do work in mice. And we have a model in which we can actually topically apply an irritant called MC903 onto mice, and they reliably develop atopic dermatitis or AD-like disease, immunologically, histologically, as well as clinically, manifesting with scaly red rash. So if we actually, in mice, perform RNA sequencing, what we find is that the cytokine and transcriptional profile of the skin is very similar to what's been reported in human atopic dermatitis. In the MC903 treated mice, we don't see this kind of profile in the control mice. But importantly for us, if we look at scratching bouts of these mice over time on the x-axis with scratching bouts on the y-axis, we find that these mice exhibit chronic spontaneous itch over time. So essentially what we're doing is we're inducing this kind of AD-like inflammation in the skin. And what we find is that in these mice, at any given moment, when you look at these mice, they are chronically spontaneously itchy. The question then for us came down to, does neuronal restricted signaling of IL-4 receptor alpha, as an example, is that alone an important target for itch? This was an important question at the time because we know that IL-4 receptor alpha is widely expressed on immune cells and probably a variety of other stromal cells as well. And so we were able to take a genetic approach where we selectively deleted IL-4 receptor alpha only on peripheral sensory neurons. And in doing so, we were able to show that there was a reduction in terms of the scratching bouts over time. Concomitantly, we also saw a reduction in terms of skin ear thickness shown here, days on the x-axis and then the percent change on the y-axis. And we also see that when we specifically delete this receptor, there's a dramatic reduction in terms of the dermal inflammatory infiltrate in this setting as well. Well, the question then became, if IL-4 and IL-13 signal through these receptors on the neuron, and this is a critically important pathway, is there another way we can choke off the itch? Well, we know that in addition to IL-4 and 13, many other cytokines, including IL-31, signal by way of their cytokine receptors to a conserved JAK-STAT signaling pathway. So in similar fashion, we thought, why don't we just go downstream and choke off IL-4 and 13 and maybe even other additional itch signals? And to do this, we actually generated in mice in which we were able to selectively delete JAK1, the one of the 4 JAK proteins, just in peripheral sensory neurons, as I showed with IL-4. And then we looked very early on in these mice, and what we found was that the level of scratching was reduced in these mice, even before we saw any difference in terms of skin thickness or histologic changes of inflammatory infiltrate. And what this told us is that actually deleting JAK1 just in peripheral sensory neurons was sufficient to uncouple the effects on itch from even inflammation, indicating that we can actually have a mechanism of action of a drug in which we can selectively target itch. So what we're now proposing is that we know that cytokines like IL-4 and 13 have clearly important effects on the immune system by way of the JAK-STAT signaling pathway, and blocking these pathways would certainly have anti-inflammatory properties. This is not really up for debate. However, what we are now proposing is that these similar pathways are conserved within the sensory nervous system, and that drugs, whether dupilumab or JAK inhibitors, or even IL-31 antagonists, may actually have neuromodulatory properties, and that may be a very important aspect of the mechanism of action of these drugs, and we could also inform the way we interpret clinical trial data or even design clinical trials. And an example here is a study we did with Insight, a phase 2 trial for a dose-ranging study, phase 2 trial for topical JAK inhibitor ruxolitinib cream in a setting of atopic dermatitis. This is a study that enrolled 307 patients across 6 different arms with both placebo vehicle, active control, triamcinolone 0.1% cream, and a dose range of ruxolitinib cream over 8 weeks with a additional 4-week open label optional period. And just real quick here, what we found was that on the y-axis, in terms of mean change from baseline in the numerical rating scale itch score as a measure of itch, what we observed very early on within 36 hours was that there was a significant anti-itch effect that could be detected at the highest doses here in yellow and red that seemed to even outpace triamcinolone cream, but certainly we see a clear separation from the vehicle placebo control, indicating that perhaps that these agents can actually have direct anti-itch effects. And indeed, in the phase 3 trials for ruxolitinib, we see effects being reported at 12 hours now. So therapeutics targeting type 2 cytokines as well as JAKs are one of the most active areas in the field of dermatology. In 2021, we'll see an incredibly just explosive year for these agents. But the point I'm trying to make here is that although a lot of these agents were developed with their anti-inflammatory properties in mind, I think what we're seeing is that somewhat unprecedented or even unexpected efficacy towards itch, and that these mechanisms of actions may actually be important in terms of understanding and interpreting these data. So what I'd like to highlight here is that we've been talking a lot about chronic itch and atopic dermatitis, and there's no question that chronic itch is very important. But what I'm going to highlight is some work by a very talented dermatologist and scientist named Feng Wang from my laboratory, who decided to take on this idea of acute itch and atopic dermatitis. And we have known from seeing patients that in addition to chronic itch, they often exhibit sudden flares of acute itch, often due to some kind of environmental exposure. This is a huge component of atopic dermatitis that's not really talked about or addressed, certainly not addressed in clinical trials. To look at this more clearly, what we did was actually collaborated with Regeneron, and we're able to look at high resolution numerical rating scale itch data on a day-to-day basis, on a daily basis. So 0 being no itch, 10 being the worst imaginable itch. They were able to actually acquire daily itch data on patients from clinical trials in the placebo group. So these patients did not get any intervention. So what does it look like when we now zoom into these patients? What we find is that there are patients like patient 1 in red who in fact experience these very severe acute itch flares, acute on chronic itch flares. So for example, patient 1 starts at a moderate NRS itch score of 4, inflects up to an 8, comes back down to a 5, goes back up to a 7, and you see what goes on in terms of the course of a month. You have patient 2 in blue who seems to have a very stable chronic itch at a level of 5. What's interesting is that although within approximately the same range in terms of moderate itch, it's very clear to us immediately and intuitively that the patient 1 in red likely has a much more morbid kind of course in terms of their disease severity, yet what's going on here is not really clear, and we're grouping these patients into the same category. So the question becomes, how many patients or what percentage of patients experience this kind of acute itch flare pattern? And when Feng Wang looked, what she found was that about a quarter, about 26% of these patients actually exhibit this acute itch flare pattern if we observe them over a month, and the others do not. The question then becomes, is this actually a subset of patients? Is this an endotype of patients with atopic dermatitis, or is this actually a much more widespread issue? Well, we actually found that, in fact, this is actually much more prevalent across patients. But what we suspected is that actually what could be involved here is that allergen-specific IgE. We know that patients with atopic dermatitis harbor allergen-specific IgE, but it's not clear, and it's been very controversial in terms of what allergen-specific IgE does. In the context of food allergy, we appreciate very well that if, for instance, a patient has allergy to peanuts, if they ingest peanut and they can become anaphylactic, that is because IgE is reacting to the peanut. Then the question becomes in atopic dermatitis, why so much IgE reactivity if it's not clear that it directly contributes to the pathogenesis of atopic dermatitis? We came to suspect that perhaps this is what is involved in these acute itch flares. Based on the kind of response and the kinetics and the environmental exposures, this would be very likely. So what we do know is that IgE reactivity is central to allergy. So atopy is defined as the tendency to produce an exaggerated IgE immune response to otherwise environmental substances. And this is mostly attributed to the mast cell. So mast cells are coated with IgE, and if in the for example, in the gut, if you have an allergy to peanuts, you would cross-link IgE and these mast cells would explode and you would become anaphylactic. There are subtypes of asthma in which aeroallergens bind IgE and then cause mast cells to become activated. The question, though, becomes in atopic dermatitis, what is the role of this? So, in fact, we actually then thought that perhaps these patients are not really a subset, but rather these patients over time, if encountering the right kind of environmental allergen, would exhibit this kind of acute itch flare pattern. So how did we explore this? In the candy cane pattern here, what we found was that if we look for an additional month, a small percentage of these patients lose this acute itch flare pattern, while an additional 20% shown here in the red acquire this acute itch flare pattern, suggesting, in fact, that there's some kind of environmental trigger that you just need to give these patients time to be exposed to. And and that's really just a matter of time that they would then exhibit this kind of pattern. So what we were able to actually do, again, in collaboration with Regeneron, is that actually look at the serum allergen-specific IgE profile of these patients. In other words, it was already known that a number of these patients actually exhibit reactivity to a variety of allergens within the environment based on serum allergen-specific IgE profiling analysis. And if you actually bin these patients into two categories, A, do they actually exhibit allergen-specific IgE or do they not? And if they do, they have there's a higher percentage with acute itch flares. So indicating that indeed allergen reactivity tracks with this kind of acute itch flare pattern that we see in patients. So the question then became which cell was mediating it. And in fact, so in the interest of time, I won't be showing this data, but we actually were surprised to find that it wasn't mast cells, which led us then to the only other cell that could possibly be involved, which is circulating basophils, which express the high affinity IgE receptor and have similar function as mast cells in terms of their ability to release histamine and a variety of other molecules. So what we actually did, and it wasn't actually shown by anyone up to this point, that basophils alone can actually are sufficient to mediate itch. So what we did to test this as a proof of concept is that we actually used mice. We crossed very specific genetic mice with basophil-specific markers such as MCPT8, in which we could then insert in a designer G protein coupled receptor only into basophils. This is a very interesting approach where we can actually insert a very specific G protein coupled receptor that only responds to clozapine and oxide, which is essentially a relatively inert pharmacologic compound, and thereby in vivo in mice we can selectively and only activate basophils. And in fact, if we do this, we see these basophils labeled in green here by MCPT8, and we can stain with avidin, and we find that when we do this, that these basophils exhibit this kind of degranulated, activated pattern. And in fact, with one dose of the CNO, and by only activating basophils in the mice, what we find is that these mice actually exhibit an acute itch flare pattern that's not exhibited within the control mice. Interestingly, then all of this would indicate there's something odd or different about basophils from patients with atopic dermatitis. And in fact, what we found is that if we actually look here on the x-axis, expression of the high affinity Fc-epsilon receptor for IgE, in fact, what we find is that patients with atopic dermatitis have a higher expression of Fc-epsilon receptor, indicating that in fact, these basophils are likely much more capable of responding to IgE in the setting of atopic dermatitis. So quantified here, statistically significant, high expression of Fc-epsilon receptor in AD patients compared to healthy controls. So what's emerging, hopefully, and is clear and very therapeutically important, is that there are lymphocytes and a variety of different immune cells by way of the production of cytokines like IL-31, IL-4, IL-13, can directly engage the sensory nervous system to mediate chronic itch. And so there's no question that this kind of interaction is very important, in a variety of different therapeutics, like dupilumab, nemolizumab, JAK inhibitors, and so on, may be very important in disrupting this axis. But what we're also highlighting here is that fundamentally atopic dermatitis is an atopic disease that involves allergen reactivity by way of IgE. And it's this inflammation in atopic dermatitis somehow primes these basophils that become much more reactive to IgE. And what we have found, actually, in the interest of time, I haven't showed this, is that these basophils, rather than primarily producing histamine, release a molecule called LTC4, which binds sensory neurons and is very potent at activating this acute on chronic itch pathway, which is a distinct pathway. In other words, there are more than one form of itch in atopic dermatitis, and allergens may be very important in this process. What does this mean therapeutically? Well, we think that actually with very potent anti-IgE therapies with the right clinical endpoints in which we actually capture acute itch flares, we may see a profound effect on these kinds of therapeutics. We could also take an approach where we selectively target or deplete basophils, and certainly we could generate leukotriene C4 or LTC4 antagonists to go after this kind of itch in atopic dermatitis. And we anticipate this will, in fact, become a future therapeutic strategy. I'd like to thank the people that did the work, the many talented members of my laboratory, former members, although I specifically highlight work by Landon Etchen and Fang Wang, our wonderful collaborators with academia as well as industry, and our various funding agencies, most notably the NIH. And last but not least, our patients for graciously being a part of our studies, whether they're translational studies or clinical trials. And thank you all for listening.